Well, here we are at week 4, the Ask Us Anything videos, and I'm not Matt Ferrari. He can't be with us today, but I will be emceeing the show for today. And we have a guest speaker who gave us one of the wonderful case studies that we had for our epidemiology week, Isabella Cadatori. And as you showed in the video, that you study interactions between parasites and the immune system, both in lab and the field. And so one of the questions from our students was, how well do your experiments in the lab translate to patterns in the field?
Right. I have to say that everything started the other way around. I spent a lot of time looking at these patterns in the field, and I really wanted to understand what was going on. And the field work couldn't really address the sort of the details of these interactions. So I tried to take everything in the lab, and we were able to culture these worms, we were able to culture these bacteria, and we started infecting these rabbits with worms and bacteria, just one worm species and bacterium, and another worm species and the same bacteria, and then everything together. And we came up with some very interesting results, both at the within host level, in terms of the immune response. We were able to characterize the immune response. Nothing too detailed, but very, in a quite generic way, but quite good results, I have to say, interesting results. And then the other thing we did was to try to quantify also the amount of shedding these animals were producing in single and co-infections. So the field work helped us to formulate hypotheses that we went and tested in the lab, and then from the lab we went back to the field saying probably the patterns we see in the field occur because of these mechanisms we see in the host.
And was that true for others, for your research? Do you find that typical pattern, field, lab, then back to field? Or do you start lab? Do you never go out of the office?
I mean, I do think it's been a huge mistake that we've tended to focus on single species and not look at the whole community, since everything that happens in you is an integral of things that have happened in the past. So I think it's a very sensible approach. It seems the obvious approach, that you can't consider one without another because the body doesn't. So why should we as scientists study just measles or malaria or something? We should be looking at the interaction of those.
I think the really key thing, though, is to make the observation in natural populations and then ask the question, how are these things coming about? And then we can be reductionist in the lab and tease things apart. Unfortunately, we often start the other way around, and we create phenomena, but we never know if they actually occur. So this is, in my estimation, this is how we should all be doing science. It's very difficult to do the type of detailed science that some of you folks have done in the field. I work in natural populations as well. So there's a lot of complications that are hard to control.
So the beauty of Isabella's system is that she was able to bring it back to the lab, and we can't do that with humans. Yeah. But there are many systems where we can't.
Certainly other animals.
So I can't do that with wolves, for example. I can't look at the co-infections between canine distemper and mange of wolves in the lab because it's extremely difficult to keep enough wolves.
In this regard, insects are really useful. Yeah. So I work on insects in rainforests and also in temperate systems with fungi to control behavior. And exactly as the case it is that we had a really beautiful pattern in nature, but to go further we had to come into a controlled environment. Typically when you want to look at the proximate mechanisms, and that's exactly what we're doing. And that, again, just like in your system, allows you to go back out of the field and better understand it. So you get a two-way flow, which is very useful. But insects are great in that case. It's better a few ants getting loose than a few wolves.
We've certainly seen your ants around. But we could also move that into a cell culture system. And I think it's a really good opportunity if we're asking molecular mechanisms. That we make observations in the field and then put them in cell culture and ask about some of these detailed molecular mechanisms. So you can do that in wolf tissue. We do it in cougar tissue sometimes. So
I think it partly depends on the questions too.
It does depend on the questions.
There are, so I work on malaria, which is mostly a human problem, at least that's the motivation. And there are questions you can ask in animal models for malaria, and there are other questions that you can't. And so it depends a bit on that. It is a big area of controversy in the field as to how good the animal models are from the point of view of saying anything about humans. But there's lots of experiments you just can't do on humans, which you can do on animals. So it has to be an iterative process, kind of like
Yeah.
Reza's been doing.
Yeah, I just wanted to jump on this. All the work on humans done on mice. And can we really go back to mice to humans?
But again,
How
I'd say that,
reliable?
that depends on the question. And I think some of the molecular mechanisms, sure, it works fine. Other issues, it's very, very different. And especially when you've got the co-infection ecology issue, that really makes
And
sense.
evolutionary questions.
And evolutionary questions, yeah.